ready to go when you are.
Okay, so thank you everyone for coming, particularly the people who have come in person. Nice to see you. And it's a pleasure to introduce David Balding, who I have known for more years than I wish to calculate.
Just over a quarter century.
Thank you, David. So David was came as professor of statistical genetics at the University of Reading when I was there. We worked there for a number of years, and then actually both ended up at Imperial, where there was another link because David was also Will Astley's PhD supervisor.
Yes.
And then David went on to UCL and then Melbourne, where he was a professor and director of the Melbourne Integrated Genetics unit.
Yeah.
So over that time, David's done a bunch of different things, mainly in statistical genetics, across quite a range. So also I remember early work on evolutionary genetics and coalescent selection, and then subsequent work, important work in forensics and in genetic epidemiology. And I think in particular, the heritability analysis he's going to talk about today has been a long-term interest. So it's a pleasure to have you speak, David. One last thing is I was shocked to find you are now recently retired.
Yes.
So yeah, I think I'm mainly shocked because that gives information on my retirement pages. But anyway, that,
it's not required. Yes, it was an early retirement. So thanks very much. It's nice to be back where the BSU, where I have many friends. There's not a huge amount of time since I spoke on a similar topic, and I've been thinking about this kind of stuff for about 10 years. And it seems to me when big problems like the forensic DNA profiling, that's generally taken me about 10 years to get to about the right place of sort of understanding it. And I feel we're in a good place now. But of course, at every point along the way, you feel you understand the problem, and then the next year you understand a bit more. So yes, this is, so as John just mentioned, I have most of my career in the UK at Reading, at Imperial and UCL, and then moved to Melbourne about 7 years ago. And as a result of COVID, the university sort of panicked and tried to push people out the door. And so they paid me to leave, and I accepted. And I said at my retirement due, I have retired to have more time for work. And I've had the most productive, my first year of retirement, I've had the most productive year, published the most papers. I'm the busiest editor of PLOS Genetics, and I'm president of the International Genetic Epidemiology Society, so I'm busier than ever. And I'm here on my way to Paris for the IGES conference, which is in a few weeks' time. With that, the basic framework is trying to predict, well, model outcomes from genome-wide data. And of course, you don't have to adopt a linear model, but pretty much most of the methods do. And to sort of set this in context, when the sort of GWAS revolution started in about 15 years ago, for a few years, we were focusing on single SNP models. And then about 2010, there was the GCTA kind of paper from the Brisbane group, which was seen to sort of revolutionize the field and realize that you can explain much more variance with genome-wide models. And basically they're using a linear, a basic multiple linear regression model like that. Of course, they're usually our covariate terms, which I'm just stripping out because I'm just thinking about the genetic contribution, but obviously that's important. And it's common to standardize the genotypes after coding them additively. You then standardize them to zero mean and unit variance. And if you do that also for the phenotype, then you don't need an intercept term in your model. And the squared of the regression coefficient is the heritability. That's just a useful language here because we don't care about the sign of effect sizes at some level. It's sort of arbitrary with the allele coding. And there are a number of reasons why heritability is a good quantity to discuss. But basically, it's just another way of talking about effect sizes. So there's two fundamental problems with this, and given the success of GCTA and sort of various genome-wide analyses since then, I think there hasn't been enough attention. People have sort of forgotten that these fundamental problems haven't gone away. One is the one familiar to many statisticians of too many predictors, Nowadays, you've got tens of millions of SNPs with imputation, and you don't have tens of millions of study participants typically, although we might get there. But slightly more subtle problem is that linkage disequilibrium, so the positive correlation among nearby markers or even not necessarily nearby, makes the coefficients kind of non-estimable because, you know, particularly if you have a lot of SNPs in perfect LD, and therefore, of course, clearly you can't allocate any signal to this SNP or the other one if they're in perfect LD, and if they're in high LD, you've still got essentially the same kind of problem. So I think it's important. So this has been quite a controversial field, and as I said, I've been thinking about it for 10 years and arguing with other people around the world, and you know, we feel we've sort of got somewhere. But part of the reason it's controversial is that there's never going to be any perfect solution to these problems. They are fundamental problems that never go away. You just have workarounds and try to make arguments that your approach is better than others, but there's no kind of gold standard proof. So to solve these problems, you have to make some extra assumptions. They're all always somewhat arbitrary. You can't estimate usefully individual beta because of the problems I'm talking about, but you can estimate heritability over sets of SNPs, and actually characterizing what you can and can't estimate is another big general problem that we haven't really thought much about, but it's pretty clear that sets of SNPs that can be quite small sets, but as long as they don't have high LD with SNPs outside the set, then you can estimate pretty well. Okay, so the ways for dealing with the problem, initially we all thought about kind of sparsity type solutions that we don't expect too many causal variants of the genome, but it turns out that the complex traits have turned out to be complex, and there are generally contributions from all over the genome. Individual contributions are often so weak that they can't be reliably distinguished from zero effect, but collectively they are important. So nowadays, and GCTA was the first sort of successful example of this, although the basic ideas go back to animal breeding, kind of blood and those kinds of things, but the GCTA sort of ushered in this, the shrinkage regression kind of approaches, penalized Bayesian approaches, where you kind of regularize the problem, you deal with the too many predictors, and to some extent the linkage disequilibrium by a penalty function, which can be thought of if it's, you know, can be a probability distribution, and therefore you can think of it as a prior distribution. So I mentioned that the sign of beta is kind of arbitrary because if you flip the allele labels, you reverse the sign. So because of that, nearly everyone, and we do as well, assumes if you are regularizing via a probability distribution, that probability distribution has mean zero and is symmetric, is not obviously true. I mean, so that's plausible if you did assign the allele coding randomly, but hardly anyone does that. And if you're systematic about coding, you know, the minor allele or the major allele as one, then these may not be reasonable assumptions, but we do make them. I just want to go back because I forgot to mention, you know, that there's a real fundamental issue about what these beta represent, and it's important that there's an important distinction going from simple linear regression for a single SNP, where it's pretty clear that the effect size is the marginal effect tagged by that SNP. It may also be tagged by other SNPs, it doesn't matter. But in a multiple regression context, the betas don't multiply, you know, conceptually, you're thinking about all the underlying causal variants, which may or may not be SNPs, that you're partitioning up their effects among the SNPs so that no signal is doubly counted. So that's a really kind of fundamentally different thing, and really you should use different notation, although we all use the same notation if you're doing a single SNP analysis versus a multiple SNP analysis because of this fundamental difference. So these betas, by definition, don't involve multiple tagging. Every causal signal is only tagged by one SNP. And of course, how you do that, there is no unique way of doing that. So that's part of the reason why the problem is kind of fundamentally, an ill-posed problem, and why there's no perfect solution. Now, of course, there's many and obviously not the only ones thinking about this. There's been many different approaches, and a lot of people have focused on different priors. This is a busy slide, but I want to say you don't have to pay any attention to this because it's just making the point that people have made lots of different approaches for different prior distributions based on mixtures of various sort of Gaussians and exponentials and so on. And there's a few pictures there, but we think that this is fundamentally kind of misguided. And the people have tended to focus on the wrong thing. So they're focused on the shape of the prior distribution, which doesn't really matter. These things are not really estimable anyway, in some sense. You have too little information. So it's kind of hopeless to think about the shape of the prior distribution. What really matters is the differences from one SNP to the next. And we do have a lot of background information to inform our prior about allele frequencies, linkage disequilibrium, from population databases or just from the genotypes in the present study, but also functional annotations from databases. And so our point of view is we don't care about the shape of the prior. Sometimes if we do need an explicit prior distribution, then a Gaussian is good enough. It, whatever distribution used doesn't make that much difference. But what really matters is the variance of the effect size. Because we've assumed the mean 0, the variance is the next obvious parameter to think about. And so basically the magnitude of the effect size, the variance term sigma squared j. So remember that notation because I'm going to be using it a lot. And it's going to vary as a function of MAF and LD. And of course, again, it's hard to actually pinpoint what's new in my talk, because many people will understand this and appreciate this, but I think its implications haven't been kind of fully appreciated in many ways, and I hope to show you some examples. So here's the kind of framework we're thinking of. We've coined the term heritability model so this sigma squared, the variance of the effect size, is the expected heritability. So I said beta squared is the heritability of the SNP. Its variance is the expected heritability. You can think about it. So you can think of it as the mean, as the expectation term in a model for the heritability. So therefore we call it a heritability model. And that's the level that we focus on. The fact that given sigma squared, the betas are then a random variable under many formulations, a random variable with that variance. The betas we actually pretty much don't care about other than through the variance term. And as I said, we don't care about the shape of the prior. It doesn't even appear in some of the models we're looking at, but the sigma squared is important and people haven't focused on it. And there's an implicit assumption I'll get to in a minute that is very widely made that sigma squared is the same for every SNP. And so that's what, that's going to be a sort of suboptimal assumption that where we do better. But so I mentioned that we can't estimate individual betas very well. We can estimate sums, and we basically fit this variance parameter. So this sigma squared is the expectation of this. So any estimate of the sigma squared is therefore by definition an estimate of this as well. For your heritability model, even like you can have 100 degree of freedom model, which for a statistician 100 parameters is a lot, but we've got so many data points here that 100 parameters is not a lot in some sense. It's no problem. The models we're going to be looking at are just regression models. The estimation is pretty easy. There are problems, of course, with correlated predictors and so on, but at some level you can have as many degrees of freedom as you like in your model, but just not 1 degree of freedom for every SNP. That's the thing that you can't afford. But with this kind of compromise, you can actually get pretty good models that give you pretty good estimates over sets of SNPs. All right, so I mentioned the GCTA paper in about 2010, which was quite revolutionary in the field for the kind of first in human genetics of using genome-wide data and finding you can explain a much more variation than with just the significant SNPs. The next big development was in 2015 using summary statistics. So I imagine many people are familiar with this LD score regression. Two plots here. So basically we fit a regression line through the association statistics, one per SNP. So this is unbinned. The regression line is still there, and it's basically doing the same thing as that line over there, but because they're unbinned, it's a bit harder to see. But when you bin them in according to the x-axis, just put them into bins, then you can see a sort of nice fit to the curve to the regression line. And the intuition here is that the more linkage disequilibrium there is, the more variation you're tagging, and therefore the more causal signal you'll pick up on average. So, in many cases, the causal signal will still be minimal, even when there's a lot of linkage disequilibrium, but on average, you'll find that the effect size is bigger with the linkage disequilibrium. But that's about multiple tagging of the same effect. And so it's a little bit, there's some kind of complicated issues here. What I'm trying to focus on are the underlying effect sizes that I said that by definition eliminate multiple tagging. So every bit of the causal signal is only attributed to one SNP. And so that's the important part of the story. So what I've written here is sort of jumped ahead a little bit and written down a generalized version of LD score regression that already gives you the kind of punchline in a way, is that the version published in 2015 had those sigma squareds here and here all constant. And so our insight was that you can actually use all your prior information about LD and MAF and functional annotations in a relatively low dimensional model for these sigma squareds, and then get much better estimates than the original. So here's I'm emphasizing again this distinction between our sigma squareds focusing on effect size tagged by SNPJ that excludes multiple tagging. So every causal signal is only tagged once, whereas in LD score regression, it includes multiple tagging. So that this is representing the effect of multiple tagging of the same signal. And so that led to a little bit of a disagreement between us and the Boston people behind this, and in part, and they were saying our intuition on the face of it seemed to be quite different, because they're saying the more linkage disequilibrium there is, the more effect you tag, the bigger the effect size. But our intuition on the face of it sounds the opposite, that we were saying the more linkage disequilibrium is, the less genetic variation there is per SNP. And if you eliminate the multiple tagging effect, there'll be less underlying heritability with more linkage disequilibrium. And I would say now that both intuitions are true. They're focusing on different things. You need to model them both. But I'm jumping ahead of myself. So there's the original LD score regression, which is just a constant times the LD score. And then our generalized version weights the LD score by this heritability model. And I'll make a claim that you do much better and show you some results. But I also want to make a claim that even though more or less everything I've said so far is kind of well known, it's been a bit of a blind spot in people's modeling that nobody's really taken advantage of it. And this list can go on forever of software that implicitly assumes the sigma squares are constant and doesn't take advantage of the fact that you can model them. And that's basically the core part of my talk. So if you've understood that, you can go home now. But I'll give you some examples and so on as we go on. Right, so how are we going to model this per SNP heritability, the sigma squared, the sigma squared terms in that the weight, the LD score? Well, the first thing we thought of, as I was just saying a minute ago, was the effect of linkage disequilibrium. And this goes back to a paper we wrote in 2012, where we talked about how there's less heritability per SNP in high LD regions. And we now kind of regret that, because linkage disequilibrium is closely related to allele frequency, and allele frequency is much easier to model and turns out to be more important. So if you model allele frequency first, the linkage disequilibrium effect is relatively small and can be just dealt with by thinning. So if you're sort of loyal fans and have read all my papers, we now no longer use much the LD weights that we introduced in our 2012 paper. It's always a problem with when people publish multiple papers on the topic that their thoughts develop. So this is a case here, and we would now have started with this model, which just says the heritability, the expected heritability in the SNP depends on its population allele frequency according to this power. Why it should be that function isn't completely obvious, but it seems to fit fairly well. You know, you might come up with a better model. We're generally dealing with relatively common SNPs, you know, let's say frequency of 0.5% or more. When you get down to very rare variants, this might not hold, and you know, we tried to do a little bit in that. And it's enough to deal with LD by just a thinning step of getting rid of some high LD SNPs. All right, in order to talk a little bit about the right value of alpha and give some intuition about it, we've got to go back and distinguish scaled and unscaled effect sizes. I said right back on the first slide that we were standardizing the genotypes, so they're scaled by the standard deviation. But in order to have a language to talk about it, so they're the betas, and throughout most of the talk, I'm only talking about these betas that are relative to standardized genotypes. But if you want to talk in general about effect sizes, you probably want to talk about unstandardized genotypes. So that's the beta star here. So this is just the usual kind of standardization. And that one in the denominator of equation 4 is basically to cancel out the normal standardization, and then the alpha value gives you an alternative standardization. So alpha equals -1 means that the sigma squared is constant. So that's the sort of standard default model that I've been criticizing, that's been pretty widely used in human genetics. And you can see here, I've already mentioned, there's more on the next slide, that we recommended about -0.25. And so there's the slide, there's the picture from that 2017 paper where we gave some evidence for this. So alpha equals -1 is this default assumption that's very widely used in human genetics. And it assumes that heritability is invariant to MAF. So that's the scaled effect size is invariant. So the unscaled effect size increases rapidly as the MAF decreases. So in order to have the same heritability in each SNP, if you've got low MAF, you have to have very big effect size. Now, meanwhile, while this assumption was almost universal in human genetics, a lot of the animal and plant breeding people were assuming alpha equals 0. So in other words, working with unscaled. So in that case, the unscaled effect sizes are invariant, but that means the heritability decreases dramatically as the MAF. So we found empirically, and this plot sort of illustrates this, so that's the uniform heritability model with the blue curve. The yellow curve is the animal and plant breeders one of unscaled effect sizes being constant, being invariant, and the red one is what we found fits pretty well. And these gray bars are illustrating just a partition of MAF into 2 parts, and showing that alpha equals -0.25 works pretty well. Back then, we had a much higher threshold for MAF of about 5%. Now we generally work with about 0.5% threshold, and the estimate is increased a little bit, as you see on the next slide. So here's some recent estimates for a number of UK biobank traits, and the average alpha value is about -0.18. And this -1, which is the default assumption in LD score regression and GCTA and all these other list of softwares, is hopeless in terms of fitting to real data. No trait we've ever looked at goes anywhere close. Even I don't think we've ever got below -0.5 in any estimate for a trait. That's really, again, the sort of simplest take-home message is that you should be including in your models the MAF and the heritability implications of the MAF. And I'll show you in a minute that you get a big boost just from that alone. But you can also do that more modeling with genome annotations. That's where I'm going to in the rest of my talk. And possibly also LD, but as I said, LD is a kind of messy one. And so slight aside that this alpha parameter is often interpreted in terms of negative selection. So the alpha equals 0.1 means that the unscaled effect sizes are invariant to MAF. So that's thought of as being no effect of selection. Now, of course, the selection can be much more complicated, of course, and one parameter can't capture everything. So this is just to sort of in broad terms, not to be interpreted too precisely, but the alpha equals -1 that I've said is the default assumption widely in human genetics corresponds to strong negative selection, and our -0.25 is somewhere in between, but much closer to 0 than to -1. And then we found this kind of curious observation that other people have reported as well, that of all the traits we looked at, having a university degree is the most selected against trait we've seen according to this measure. So it seems a bit sad that nature should hate education that much, but and of course there's sort of various stories about why that might be true, but I'm not going to speculate too much. So here's some example that it helps gene-based association. Actually, we had this idea for this paper about 8 years ago, but we've only just finished it for various reasons, and it's under revision. It's on medRxiv, and it's being revised for a journal which we're hopeful will appear soon. So just focus on SNPs in a gene, and obviously define genes however you like. And there was something came from Jennifer Liscarton and colleagues nearly 10 years ago, a gene-based association test based on kind of linear mixed model ideas. And I'm not going to say much about it here, but our genome-wide linear predictor is closely tied in with linear mixed model kind of ideas. And so they were only thinking about individual level data in 2013, and they developed a sort of likelihood ratio test of the heritability being 0. But what they didn't have was this subscript J, so they didn't have the heritability varying with SNPs, and so that's our generalization. So we do much the same thing, but based on but including more general heritability models rather than the uniform model, and also we've extended it to summary statistics. But the kind of mathematics of that is complicated enough that we needed to have a fairly simple heritability model. And so we just use this simple MAF scaled heritability model after thinning to deal with linkage disequilibrium. So that's the model I was describing in the previous slide. For whatever reason, even though I'm an author on this paper, I didn't really agree with this label. They called it the human default model and of course we could potentially do better, but there's a little bit of complexities. But we've just got this very simple heritability model, and it helps us to do quite a bit better than other methods. So these are various other methods for gene-based association. I won't go into any details here, but if you're familiar, some people might be familiar with some of this software, Magma. Actually, it turns out that one of the reviewers of our paper was one of the authors of Magma, but he turned out to be quite nice and didn't said positive things in the review, which is good. But we did much better than all these other methods just based on that NAF adjustment in the heritability term. Over here is comparing our method with just a min-p kind of single SNP analysis for the same sample size as that first blue bar. And then if you increase the sample size by 20%, that's the second blue bar, and then increase by 40% is the third one. So you see it's somewhere around about corresponding to a 30% improvement increase in sample size just by using this heritability model. And finally, in the bottom right panel, we tried this for different values of alpha. So these are detections from real data, so it's not actually power, but we've shown that the model has good type 1 error. So therefore, we're sort of confident that more detections means more power. And we found again that alpha being -1/4 works best. There's alpha equals -1, which is considerably inferior, but also 0 is also a little bit inferior as well. So there's an explicit example where modeling this heritability and not just by default assuming the same heritability for every SNP gets you some gains in gene-based association. I'll talk about prediction a little bit later, but for the prediction paper, we used a more complicated model. So I've got to go into that a little bit. Once you've dealt with MAF and LD, the next thing you think about for predicting heritability of a SNP is the genome annotations, functional classes and all that kind of thing. So the Boston group behind LDSC very quickly cottoned on to the fact that with this simple regression model, you can afford to put in lots of predictors. And so they came up with 24 functional categories, just binary indicators of coding or non-coding and intronic and not intronic and lots of things like that. DNA's hypersensitivity, I'll come back to them in a little. So they then developed this 53 parameter model, and as I said, for statisticians, 53 sounds like a lot of parameters, but it's not no particular problem here, either to compute or to estimate them. But they also developed a little bit further to allow, rather than just binary predictors in the model, quantitative traits that could be proxies for LD. And they came up with this baseline LD model with 75 degrees of freedom. And by then we were sort of been arguing with this Boston group about various issues, but they definitely had to jump on us in following up this idea that was obviously the right way to go. But in the context of all these arguments, we had various kind of ways of trying to show that our model performed well, but we didn't have anything unequivocal. What you really want, of course, for model comparison, ideally, is a log likelihood comparison. But this is a log likelihood for summary statistics. So we did develop such a thing. So marginally, the association statistic has a chi-square 1 distribution, and it's you can express the mean as a function of so this is not necessarily null, this is a non-null distribution. You can express the mean as a function of the heritability. So this is where the heritability model is going to come in. The difficult thing is that the fact that the test statistics are correlated because of linkage disequilibrium, we just did something simple of dividing by the sum of the R squared, something like the LD score, and we did some tests to kind of persuade us and hopefully others that the likelihood approximation works pretty well. I won't go into any details. That was in our paper. That's all published work if anyone wants to know more details. But just to say, we came up with a log likelihood approximation that gave us the ability to compare models, we think, pretty accurately. And so there's a number of different models that from different groups around the world, and some have allele frequency in them and some measures of linkage disequilibrium and various measures of genome function. And when we looked at it again, as I said, the Boston group, having developed these models with genome function in them, did much better. So we were able to compare models. We got the bad outcome that our model wasn't competitive with their model, but we turned defeat into victory by basically stealing their model and making it better by getting rid of, they had a very crude way of dealing with MAF by just 10 bins with a separate degree of freedom for each bin, which is pretty crude and not very good. So we put in our better allele frequency model. We did throw our LD weights in as well, but we wouldn't do that if we were doing it again. We think that they're not quite good enough. So we then came up with this BLD LDAC model, which is a sort of prior model for it's prior in the sense of before you, let's not even think about priors. It is just a model for the heritability of SNPs, which you estimate from the GWAS. You estimate parameters from the GWAS data. And in this case, our model had about 66 degrees of freedom, depending on, you could either estimate alpha, which greatly increases the computational burden, or you could just use this minus a quarter value, which we found worked pretty well. So I gave you a DAG earlier. Here's a sort of slightly more complicated DAG. I shouldn't have to tell people here that it's actually a bit surprising. It was a long time before we actually wrote down a DAG, and it is actually quite illuminating that even though it's a fairly conceptually quite simple. So again, we don't have to worry too much about the details. The taus correspond to the functional annotations. The alpha was the MAF parameter. The A is the intercept in LD score, which is when the LD score paper was published, it was largely promoted as an alternative to genomic control and correcting for confounding. So some other work we've done says that doesn't work very well. And basically you've got to have other evidence that there's no confounding due to population structure, or you're likely to be led astray. So you can't rely on this intercept, although it might help a bit. Right, so that's a sort of schematic view. And again, we focus on this sigma squared. We don't really care about the betas directly. And as I said several times, we can't really estimate them, but we can estimate these sigma squareds with a model that has degrees of freedom much less than the number of data points. So now on to prediction. So this is a paper by Doug and colleagues in Aarhus. I'm not an author on this one, but it's sort of closely related with the work I've been talking about, and another example of how you can make big gains on standard methods. And I think this is quite impressive. So there's two slides here. The first one is for individual level data. And so Doug basically took existing methods for predicting. So that line right across the middle there is a kind of standard PRS prediction. These various other methods, Bolt, LMM, Bayes-R, ridge regression, this is a version of the lasso, and they all do much better than a standard PRS, which is no surprise. But each of those methods can be improved by, just like we did for LDSC, you generalize it by putting in a more general heritability model, because they all implicitly assume this uniform heritability. So the dark blue is our BLD LDAC model, which at the time of our 2020 paper we thought was the best. Again, we've moved on a bit since then. But the light blue is this LDAC thin model, which is basically, I've cut this figure out from the paper, so it's the term we used at the time, but it's actually, I should call it I've called it the that's the MAF model that I introduced a couple of slides ago. And I think that would be a better name, because it is basically the MAF model, and the MAF is the main term. Plus we also thin. The thinning is relatively unimportant, but just to get rid of a few high LD SNPs. But you can see with that simple 1 degree of freedom model, you already get big gains over these existing methods. And then for an extra 65 degrees of freedom, you get a little bit more. But obviously you don't need all those 65 degrees of freedom. So I'll come back to that in a minute. These are all, so LDAC is Doug's software, so it can be slightly confusing. So Doug's got a whole very big and sophisticated software package called LDAC. It was originally LD adjusted kinships is where the term came from, because we were thinking about kinship matrices in mixed models at the time. We moved away from that over the years. And he re-implemented all these existing methods, the Bolt, LMM, and Bayes-R, and he's a bit of a whiz with computing. So his version of Bayes-R is 50 times faster for this analysis than the published version of Bayes-R. And the Bolt LMM is also faster than the published Bolt LMM. And obviously these are just ridge regression and lasso, but they're quite efficient implementations. And the average R squared again corresponds to something like a 25% increase in sample size. I think that's for our BLD LDAC model. So this relatively simple innovation of generalizing the heritability model gets a pretty substantial gain. This now is the same idea for, one more thing to comment on here, that of those 4 methods, for the individual level data, we found the Bolt LMM was the best. And so this 25% is relative to Bolt, which was the best of the existing methods. And that's the gain we got by generalizing that method to include these better heritability models. But with summary statistic data, we found Bayes-R was the best. And so these plots are giving the same information in slightly different version. Again, the LDAC, the light blue is the LDAC thin, the 1 parameter model that just has allele frequency only. And then the blue is the 66 parameter model that we thought was the best at the time. And again, the story is the same. You get most of the benefit just with the 1 parameter model, and you get a bit more with 65 parameter model. Unfortunately, you can also see here that our biggest relative gains are when the prediction is poor. And that's what the binning over here is to show. And unlike for individual data, we always did better. But here with summary statistics, we don't always do better. There's 2 points below the one where we actually did worse with the 1 degree of freedom heritability model. But you can see the 223 out of the 225 traits, we did better by improving the heritability model. And here you get some idea that for these poorly predicted traits, we get a sort of 15 to 20% improvement. And for the better predicted traits, we get a lower relative improvement of 5 to 8% or something like that. Right, how are we doing? We have the problem that all many statisticians face at one time, that you want to compare yourself with existing methods. And therefore, we kind of stuck with these functional categories that the Boston people introduced about 5 or more years ago. So the more recent work, we've updated all of this. But here's a set of fairly standard functional annotations of the genome that's a little bit out of date, but broadly speaking, things like coding probably haven't changed very much. Is conserved in primates. Again, there's a specific definition of that, to give you some idea. It's about 2.5% of SNPs lie in conserved regions. The DHS, so there's fetal DHS. Oh yeah, there it is. DNA's hypersensitivity sites. That caused a bit of a splash at the 2018 American Society of Human Genetics conference, when the Boston group announced that they found a huge amount of heritability in these DHS sites. And I'll show you in a minute that when we reanalyzed, we found that that effect completely went away. And as is common in the scientific world, they never said they were wrong. They just never repeated the claim. So what does turn out to be important? So these models do kind of 2 different things. One is that these can all be predictors of heritability that can improve your model for prediction or association, as I've been discussing. But you can also analyze the parameters associated with these models to talk about, which are the biggest contributors to heritability. So what do you think would be the biggest contributor to heritability out of this set? There's a very clear and unequivocal answer.
Coding.
Sorry?
Coding.
Yeah, you might have thought so. And in some measures, yes, but because coding is fairly rare, no, conserved is way ahead. And I'm a bit puzzled about, so I'll jump ahead and get to that since I started on this. So here's 2 measures of the effect. The first one is is something which has come to be called enrichment in the field, which is how much heritability there is in this category relative to some kind of baseline. And what the baseline is a bit dispute, but what the Boston group first did, and we've ended up going along with, but after a fight, is the baseline is just proportional to the number of SNPs in that category. And so there's 7.5 times as much heritability in these conserved sites than you would expect under a null model. And you can see all these categories that you would expect would be important: transcription factor binding sites, promoters. So when you look at the top plot here, they're all enriched for heritability, as you would expect, but probably less than you might think for many of them. But this bottom plot is the regression parameter, and you can see the regression parameter is often not significant, and it's not big for anything other than conserved. And the difference is, well, one of the key differences, this enrichment is a kind of marginal thing, just only looking at this category. How much heritability does it have compared to a null model? But this is a joint, this is a joint fitting of all of these parameters. So this is saying how much, what's the effect size of being a conserved variant, given that all these other variants are in the model. And conserved is way out here as being very much important, even though we have all these things like coding and 3' UTR and everything else is already in the model. So what's causing conserved to be so important isn't any of these things, apparently. And it's a bit puzzling. I come from a sort of math stats background. I've not really got the expertise to go any further with this, but it just seemed curious. And there was another interesting thing with DNA's hypersensitivity sites, which were claimed by the Boston group to have this huge enrichment of explaining nearly all non-genic heritability they claimed at the time. We found only a modest enrichment of 1.7, whereas they had reported 4. But interestingly, the regression parameter is negative. So conditional on all the other terms in the model, the effect of being a DHS SNP is negative. It reduces your expected heritability, which again is a little bit puzzling because people would have thought that, given the understanding of what DNA's hypersensitivity sites are all about, we would expect them to make a positive contribution to heritability, but they don't according to our analysis. Coding is second, but yeah, way behind conserved. And then not much else is even significant. There are some epigenetic marks that are significant here, and a couple of other estimates are negative. I think DHS is the only one that's significantly negative. Oh, except for this. Yeah, no, actually, I think it is the only one. All right. So This slide just gives a little summary of things that I've already told you, that the most functional categories are enriched, but the corresponding regression parameter is often not significant. So this is all about overlap with different categories and so on. Conserved is by far the biggest effect. DHS is interesting because it's enriched a fair bit, 1.7, but the regression parameter is negative, which is a little bit puzzling. I've got something here that I mentioned about the effect of the baseline might be important for conserved, because conserved sites have relatively low variation, so they have relatively low number of SNPs, and so that might affect, so that obviously affects the baseline. That might be important in ways we haven't really explored. I just go back to the slide I skipped over, which is this alpha parameter that you can think of broadly as in measuring negative selection. So you would expect all of these important functional categories to be somewhat resistant to mutation. And so indeed, that's what we found. These alpha parameters are significantly more negative in these regions, in each of these regions, than outside them. So in other words, you get more negative selection if you're a coding SNP than if you're a non-coding SNP. That shouldn't come as a big surprise, but it's also true for all of these other categories here. Out of 24 categories, 18 turned out to be significant, 17 of them in a negative direction, but this repressed category in the opposite direction. So these kinds of analyses have been done by other people, but I think not as thoroughly, and we think we've got better models, and so we think our answers are more reliable. I've gone some way to convincing you of that. But I've already mentioned that this 66 degree of freedom model is a bit ridiculous in having so you've got all these non-significant regression parameters, but it's probably because it's, an over-parameterized model and lots of correlated parameters. So let's see if we can slim down the model a bit. And I've had a PhD student working on this and came up with a 10 degree of freedom model, but actually you can even go down to 3 degrees of freedom and do almost as well. And so here is just some analyses. Again, these are sort of standard analyses that other people, so looking at out of sample prediction models trained in the white British UK biobank and then predicting in the South Asians and East Asians and Africans, this is all UK biobank data. And so the 10 parameter model is virtually indistinguishable in performance compared to the 66 parameter model. And similarly, this is trained and prediction in the Taiwan biobank. And there's a kind of similar story that the 10 degree of freedom model is almost as good as the 66 degree of freedom. I won't go into much more detail here, and again, these are all analyses that we've been able to do that other people have already done, but we think we've got better models and our results are a bit more reliable. So here's male versus female heritability with some significant differences, 25 significantly different out of 196. So that's for the total heritability. Genetic correlation is looking at something quite different, but we found 80 of the traits, so not far below half, have genetic correlation between males and females, significantly less than 1. So this is all saying that, obviously the genetic architecture of males and females is quite similar, but there are, differences that may be relatively important for some analyses. This is a measure of EQTL, the enrichment of heritability in EQTL sites, a broad definition from the GTEx data. If it was significant in any, if it showed up as a significant EQTL in any tissue type, then we class it here. So it's a fairly common category of being an EQTL. I think the baseline was about 6% of SNPs were EQTLs according to this definition. The overall enrichment on average over a number of traits was about 2.8 for these EQTL sites. So they were enriched, not surprisingly, but again, maybe not as much as you might have expected. There's some summary of things that I've already talked about, so you can have a quick look and remind yourself. I don't think I'll say much more, except where to from here. I want to make the point that what I'm selling is the general framework. None of these models is particularly the last word, and partly because we're working with slightly outdated genome predictors and this kind of thing, and there's a whole lot of interest in all these predictors of functional sites and composite predictors. So we saw recently Macy from Jiong Lin and colleagues at Harvard, which is a composite predictor with three degrees of freedom, so that can fit quite well in our framework, and we have some idea that from preliminary analyses that it might perform a bit better than the models we've looked at, and there are lots of other predictors out there. So I don't think any of, and we've got our own new 3 degree of freedom model and the 10 degree of freedom model. So there's a bit of jostling around, but once you've got the basic idea and once you've got the MAF term in there, the gains beyond that are not so great, but there are some gains to be had. So I mentioned I've had a very productive time since retiring. I wrote this, we wrote this paper now out in bioessays, which summarizes a lot of what's in this talk, which is a sort of review essay. So we basically, try to give a broad picture of this general field, some of the work at the end, the 10 degree of freedom model is a just submitted PhD thesis, not yet examined. This is our medRxiv paper on the gene-based association, and that's the prediction paper that I'm not part of. So thank you, and I will stop there.
Thank you. That was fun. So we have time for a few questions. Should we try the online people first, or do you want us to? You've got any?
No one yet. Oh no, sorry. Hands clapping. Hands clapping. Yes, if you have a question online, please do raise your hand.
Well, let's see if we can warn them what we want from the room there.
Yeah.
So Will.
Not very sophisticated question, but I was just wondering about the sex differences in heritability.
Yes.
Is that got any biological interpretation? Because I was thinking, presumably you could have a situation, for example, where the difference in sex caused a multiplicative change in your phenotype, and if you log the phenotype, then you get an additive change. You might not see any difference on the log scale, but you might see a difference on the.
Yes, I hadn't thought about that aspect, but there was quite a, and I've just forgotten the author now, but a paper that got quite a lot of attention a couple of years ago, just making the general claim that there are important differences between the sexes in heritability, and all modeling in human genetics should take account of these differences and have separate models for males and females. So it's, as I said, a lot of this stuff is not completely new. This is analyses that other people have done, and we think we've got a better version of the analysis. I hadn't thought about the point you're raising, but I think it's pretty widely accepted that there are some important differences. And as I said, I have another picture that I didn't show here that I could have, that shows the traits that have relatively low genetic correlation between males and females. And they make a bit of biological sense to a non-biologist such as myself, body morphology kind of differences. And I think there's something to do with wrist strength or something like that, that you sort of think that sort of things that might be different between males and females. I don't know. But I think it's pretty widely accepted that there are some differences. Whether the differences are big or small is a matter of judgment, right? In general, the genetic correlation between males and females is close to 1. And it's indistinguishable for the majority of traits, but for some traits it goes down to below 0.8, so I think that probably is indicating some differences in genetic architecture between males and females that may be important for some analyses. And in fact, all our prediction stuff, I can't remember now whether we took account of sex differences in the prediction, surely in predicting. And then we didn't predict height, because then you would want to take that into account. But broadly speaking, you probably want to take sex differences into account in your prediction, I would have thought.
Sorry, I should have asked people to say who they are before.
So that is Tina, postdoctoral group in American. I would like to know, when you say the conserved SNPs
Yes.
are a good predictor of heritability,
Yes.
are they distinguished between coding and non-coding regions, or both are regions or not, or are they whole genome included?
It's the whole genome is included, so it is just looking at whether they're conserved across certain I think, is it mammalian species or primates? I've just forgotten now, but it's, it's comparing human genome with at least primates and maybe other mammals. I've just forgotten now. And so the definition is, I think, agnostic as to any of these kind of functional categories. So they're not just focusing on coding or any other genome category. It is genome-wide, I believe. And obviously, it's a bit open as to how you define, how much conservation, because it's a binary indicator, whereas obviously you'd rather have a quantitative variable to measure conservation, but we didn't have that. I mean, there are other measures of conservation around, but this binary indicator just came out. So as I said, it's about 2.5% of SNPs were categorized as conserved, and that did just come out to the most important predictor of all those functional categories. Yeah.
Do you have any measure of if they are in a different region in the genome?
I do have overlap information. It's in the PhD thesis I was talking about. If you want to come up, I've got it on my laptop. I can see if I can show you, but we do have some overlap. So for example, we were puzzled by this result with DHS. They're enriched for heritability, but the actual regression parameter is negative, which seemed a bit puzzling. And so we did look into that. The DHS sites overlap with a lot of other sites which have higher than average heritability. And so it's DHS are enriched because they overlap with transcription factor binding sites, I think, were one of the key ones that are overrepresented in DHS sites, and they carry high heritability. So when you take that effect out, because you're jointly modeling them in a multiple regression model, when you take out the effect of transcription factor binding sites and a couple of others that were important, then just being a DHS site doesn't convey extra heritability, in fact, significantly less.
I was going to ask you about that, because that was one thing that struck me, in that a DHS site is kind of a bit of open chromatin.
Yes.
In some cell type, the cell type specificity probably matters.
Yes.
But also it's kind of the price of admission to be functional.
Yes.
So it's like it's a DHS and not anything else.
Yeah. So when the Boston group announced that nearly all non-genic heritability was in DHS sites, that seemed kind of plausible exactly for those reasons. So why we came up with a negative, it is a bit puzzling. But we did look into it a bit, as I was saying. You can explain it in terms of the representation of these other categories in DHS regions.
I think that makes sense in that if it is only DHS and not any of those other things, it's probably not doing anything. But I'd be interested in whether, have you looked at something like the putative regulatory element definition from ENCODE?
We have, in that later work from my PhD student, he looked at all kinds of variables from ENCODE. I said he started off with 335 predictors to get down to his 10 predictor model. And so we had lots of ENCODE annotations included as potential predictors. I've just forgotten whether they came out again. I've got his PhD thesis here. I can have a look maybe later when we're having a drink. We can go over that. But yes, and as I mentioned, there's been a huge interest in these kinds of predictors of function, which is more or less the same thing what we're talking about. And so there's lots of new predictors available. And also partly our observation about DHS, which is a bit puzzling, it's partly because we have, in order for direct comparison, we've been using the same definitions that the Boston group started using more than 5 years ago. So there's probably, to some extent, they're out of date. And we're now definitely ditched those, and in future work we'll be using more up-to-date definitions. But for that analysis, we were still using the somewhat out-of-date functional categories, and so probably the data is better quality now, I would guess.
Steve.
Okay, Steve.
Yes, Steve Burgess, MRC, BSU. How much could relatedness between individuals, could that influence your heritability estimates?
Yes, so I didn't really go into this, but in general, of course, GWAS studies generally exclude related individuals. So there is a kind of assumption that we're dealing with, broadly speaking, unrelated, although of course we're all related at some level, but broadly speaking, unrelated individuals. Yes, I think any substantial amount of cryptic relatedness there would lead to genome-wide linkage disequilibrium, which would be there are diagnostics to check that is not a problem. So I don't think it is a problem for our analysis, but yes, broadly speaking. And the problem of population structure, I mentioned that, confounding the population structure, nothing I've said here eliminates that problem, but it is a kind of standard problem, and there are diagnostic checks you can do to sort of convince yourself that it's not important, and that's, yeah, that's basically, looking for genome-wide correlations in different ways.
Have you considered going the other way around and comparing to within SIB analyses?
That is quite interesting, but I haven't. I wonder if we would have anything useful to add, but certainly you can, within the sort of framework I'm talking about of genome-wide multiple regression, would that work for SIB pairs? I haven't thought about it. It's not obvious that we'd be using that kind of modeling setup, so I'm not sure that it could do it. But that is an interesting kind of approach for lots of reasons. And similarly, things like in admixed populations of looking at, categorizing people, measuring different levels of admixture, and so that's kind of, varying amounts of genome sharing, and so that's a bit similar to the idea of varying amounts of genome sharing among SIBs that can be informative. But in any case, we haven't done any of that. But,
Can I just ask one more?
okay.
So sorry, I should have introduced myself. I realized so John Whittaker, I'm director at the BSU. So the varying heritability with allele frequency. One of the things that made me think about was a paper from Gonzalo's group a while back, showing that the false discovery rate of associations in rare variants was substantially higher than that in common variants, which is exactly what you'd expect if you use the same p-value.
Yes.
So he had a clever but somewhat complicated resampling scheme to try and adjust for that.
Okay.
Recall. Have you thought about trying to produce like significance thresholds that control FDR, like stratified by allele frequency or dependent on allele frequency?
No, that's true. That would be an interesting thing to do. And no, we haven't. I mean, generally, as I mentioned, we've been focusing on relatively common variants. The threshold we generally use now is 0.5%. And whether that power relationship still holds as you go down below 0.5%, we don't know. Obviously, it's hard to measure, but we should have enough data these days with genome-wide data in UK Biobank to start fitting those models and get an idea about that. We do already have a bit of an indication that when we went from a threshold for 5% down to 0.5%, our estimate of that alpha parameter increased a little bit from 0.25 to a little bit higher than -0.2. And so but then it's hard to tell whether that's some kind of dilution effect due to the lower information content, or whether it is indicating a change in relationship as you get down to the rarer variants.
I guess you've got more imputation error.
Yes, yeah, that's the sort of thing I was thinking about, that you've got lower data quality, and so that will tend to maybe shrink the parameter towards 0.
Yeah, my other memory of that Gonzalo paper is actually he was in the same kind of allele frequency as you're in, so that effect was seen even like the low findings.
Yeah, okay.
So we do have a question from Mark Bailey on Zoom. Mark, would you like to unmute yourself and say your question, please?
Thank you very much indeed. Just to point out that I'm an interloper from University of Glasgow. I hope that's okay. So David, you said near the beginning, you were talking about assumptions in the early models that the distribution of effect sizes was symmetrical around 0, based on the assumption that the reference allele has been selected at random. So given the way the reference genome was put together, it's quite likely that for common SNPs, the reference allele is not quite randomly selected in that case.
Yeah.
Do you think, so is that still an issue, and is that something that has been accounted for fully in your math scaling in your models, etc.?
So no, that is one of the many kind of gaps here that we haven't really looked at. For that particular thing, I didn't say just the early models, all models that I'm aware of, every other group that I've seen doing these kinds of analyses has assumed when they've put a prior distribution on the effect sizes, they've assumed it has mean 0 and is symmetric. Everyone does that, and we do it. And it may not be true exactly for the reason you said. It would be true if the reference allele was assigned randomly. But as you say, in general, it's not. We kind of think, so, it's possible that for some traits, the rarer variants have, a slightly, hang on, what is it we're saying? The effect size, yes, if with reference to the rarer variant might be, well, it might be systematically, yes, non-symmetric.
Yes, systematically unsymmetrical because of the effects of negative selection, etc.
Yes, exactly. So now we think that the effect won't be very big, but we haven't, that was one thing I was trying to get my PhD student to do, but he had enough other things and he never got onto that to investigate it. So as far as I'm aware, nobody's looked at it. It wouldn't be too hard to do. I mean, one thing, which is actually what I suggested my PhD student do, but he never got around to doing it, is to actually redo the analysis after randomizing the allele coding and see if the answer changes. That would be a sort of a bit of a robustness check. And I suppose you could, yeah, it depends on, so if you're playing with UK Biobank and you've got access to the genotypes, of course, you can create your own summary statistics, but if you're dealing with summary statistics from a GWAS, you don't have much flexibility over this. They might tell you what the reference allele is, but yeah, I'm not sure how we can explore that. But in any case, I agree that is a bit of a gap, and we're kind of guessing that's not very important. I mean, another thing I meant is that I did see a paper on this that got onto arXiv, and I never saw it appear in print, that people were arguing that these underlying true effects after eliminating the effects of multiple tagging could be correlated. And they were arguing that there is a negative correlation of effect sizes at causal variants that are close to each other in the genome. The more LD there is between the causal variants, the stronger the negative correlation in their effect sizes. And so they tend to cancel each other out a little bit. And there's some evolutionary kind of argument why that might happen. But we don't, I'm a bit skeptical that that effect is important, and it'll obviously be just pretty low range, but we assume, again, like everybody else, that these effect sizes not only have mean 0 and they're symmetric, but also that they are uncorrelated. And none of those things likely is strictly true, and we haven't investigated them. But again, going back to the fact that effect sizes are so hard, are sort of essentially inestimable at some level, it's not entirely straightforward to check.
Sure, thank you very much.
Question on the, I should say, a continuing job from the statistical laboratory here in Cambridge. I have a question about the choice of the normal distribution as the prior.
Yes.
As in the, like in fine mapping, when we want to locate the causal variance, usually assume the distribution of beta is like a spike and slab kind of distribution.
Yes.
But here you assume it's normal.
Yes.
I guess there's also a heritability model there that can be used as well.
Yeah.
So could you comment on that difference?
So for genome-wide models, we've done some pretty big kind of robustness checks and really changed the shape of the distribution and found that, and in simulations, and then found that it had practically no effect on inference. So we reckon that for genome-wide analyses, that's the analyses are pretty robust, and my general intuition for this is that you have so little information about each effect size that shape of the distribution. But if you're doing fine mapping and you're focused on a particular genome region, then it might be more important. The spike and slab type models, I somehow can't think of them as being realistic. That's a kind of practical assumption that by assigning 0 effect to certain variants, you basically simplify the interpretation of your model, right? Because they become the 0 effect variants become kind of ignored. And so that might be useful as a practical measure, but I just can't believe that that's realistic. And I would have thought some kind of smooth curve would be more realistic. I mean, if you're talking about a literal spike with an atom probability of 0, is that what you mean, or just a sharp?
Yeah.
Anyway, so I don't think it matters for genome-wide analyses. It might matter more for for fine mapping because you're focused on
So would you
particular
use
region.
your heritability model as a prior in gene climate?
Well, for example, in our gene-based association, we I don't think we kind of checked that, but for our individual level analysis, we did assume that Gaussian prior, but unlike most others, you know, nearly everyone else assumes the same variance, but we, you know, that's our key innovation, is to allow the different variances. But then we derived a summary statistic version of that, which doesn't have any distribution for the effect sizes. It only has the variance and no other feature of the distribution. And we did some comparisons to show that those two approaches were quite consistent with each other. So that's a bit of an argument again that the shape of the distribution doesn't matter, because the summary statistic analysis, there's no explicit role for the distribution of effect sizes, only the variance of the effect sizes. But it's not really. So we're pretty convinced that it's not important, but we can't be absolutely certain. We haven't investigated it really thoroughly.
What can you tell someone? I think we should stop there, and I think David's going to be around for a bit longer, and also we will be going for a beer afterwards. So other people who came in person will get further opportunities.
All people online at the Cambridge Blue Pub, right?
Indeed.
Yeah.
Yep.
So, yeah.
So should we thank David again? If he's going to break down, he breaks down the road.
Yes. Sorry. Yes. Yeah. It's going to break down anywhere. It will be break down for rare variants. Yes, and we definitely know that there are big effects.